Yes, good morning, Samara. Can you hear me?
Yes, sir.
Okay, good. So, yes, so let us start. So, have you gone through the details?
Yes, sir.
Okay, very good. So, let us, I'll just review it today. Let me just open
Okay,
the
sir.
presentation.
Sir, I have to ask you certain questions.
Yes, you're most welcome. You're most welcome.
Okay, sir.
Yes.
Okay, sir.
First you ask question, then we'll start.
Okay, sir.
Yes, tell me.
Sir, there was a point named retention factor.
Yes.
Sir, can you please explain me?
So retention factor is like, you know what is retention?
Sir, to retention is to retain it for a while.
Retain means like to hold, right?
Yes, sir.
And in
Yes,
chromatography,
sir.
everything depends on retention. Like the separation depends on retention. If there is no retention, there is no separation, right? But if there is very high retention, again there is no separation, right? Yes, Samra.
Yes, sir.
Yes. Can you hear me?
Right, sir. Yes, sir.
Okay. So retention factor.
Hello. Yes, sir.
Yeah.
Yes, sir. Yes, sir.
I'm there. So
Yes, sir.
retention factor is like it is used in, initially it was used in TLC, and later on it was extrapolated to all other type of chromatographies. So retention factor, or we call it relative front also, right? So retention factor is,
Yes, sir.
it is denoted by the word RF. RF value you might have heard
Yes,
of
sir.
in TLC. So RF value is distance traveled by a solute divided by distance traveled by a solvent, right? Now, if you
Yes,
just
sir.
see this slide, I'll show you one slide of, sorry, we are going down. Now you see here, how do we know that the separation has taken place or not, or the separation is good or bad? Now this is one plate which I developed in my laboratory for amino acid paper chromatography. And you know, there are two distances. One is called...
Sir, excuse me, sir.
Mm-hmm.
Sir, I can only see the first slide.
Okay, you can only see the first slide.
Yes, sir.
That means again, internet is not very good at your place.
Yes, sir. Now I can see.
Means that is you're able to see it when it is in the open mode, but when it is on the screen share mode, then that means the slide is not moving at your end. Can you see it
Yes,
now?
sir. Yes, sir.
Okay, now you see there are two front. One is called solvent front, another is called solute front. So what happens actually? So this is a TLC plate. This is ascending TLC. When we dip this TLC plate in a solvent, then I think you might have read about these things. Now, this is a TLC. So how it works actually?
Yes, sir.
We take a paper or a TLC plate, we dip it in a solvent. That solvent is named as mobile phase. Now the solvent will rise due to capillary action. And we have a point that is called loading point here. This is loading point. You can see this is loading point. So from
Yes,
that
sir.
loading point, the samples, they start moving upwards according to their polarity again. Like mostly I tell you, like when we are using TLC, we are using silica as a stationary phase. So when you are using
Yes,
silica
sir.
as a stationary phase, silica is polar or non-polar?
Sir, silica is polar.
Polar. So if this is a silica plate, I think you can predict that this compound, where is like the top compound, my cursor is here. Can you tell me whether this compound is non-polar or polar?
So which one?
This one, my cursor, you can see the top compound here in this slide. Can you see the slide?
I cannot see the cursor.
Oh, you cannot see the cursor.
I can see
Okay.
the slide, but
You
yes, sir.
you can see the step 1 where my cursor is.
Step 1. Sir, in that figure, the TLC of aqueous extract of terminally Arjuna.
Okay, you have. Okay, chale, okay, you have. Because again, the problem is that slide is not moving at your end. Now you see it. Now
Okay,
it is
sir.
moving, I think.
Yes, sir.
So that means
Okay,
due
sir.
due to
Yes, sir.
poor internet connection, the slide is not moving. Now you see it is step 1. Can you see my cursor
sir.
here now?
Yes, sir. Yes, sir.
Now see there are like the sample was loaded here and you have so many spots, means every spot is representing a compound. Now one compound
Yes, sir.
is here on the top, the plate is silica plate. So tell me that whether this compound is polar or non-polar.
So 1 minute.
Yes, yes, take your time.
Sir, it's the polar one. It's the polar.
It is polar, but your answer is wrong actually. Because if it is polar, the polar compound will have more interaction with the silica because silica is polar.
Yes, sir.
So the
Yes, sir.
polar compound will have less migration, right?
Okay, sir.
And the non-polar
Yes, sir.
compound, the non-polar compound will have more migration.
More migration.
So a compound which is polar will be here. You can see my cursor. It will be here near
Okay,
the
sir.
loading
Okay,
point.
sir.
And a non-polar compound
Okay,
will
sir.
be on the top. Now you come to this slide. So this line is called loading point. What we call it? Loading point, sample loading point, where we are loading the sample.
Okay, okay, sir.
Then we draw
Okay,
one
sir.
line on the top. This is called solvent run mark. Up to this
Okay,
mark
sir.
only we allow the solvent to run, the mobile phase to run. So this is the maximum distance up to which the solvent is running. The solvent is always said to be the unretained species in a chromatographic system. So what do you mean by unretained species?
Sir, the one that is not retained.
That is
on
not
the stationary
retained
phase and moves out. Yes, sir.
And moves the fastest. Okay.
And that, yes, sir. Yes,
The
sir.
solvent will move the fastest.
Fastest. Okay,
Because
sir.
why
Yes, sir.
fastest? Because it is said to be the unretained species.
Unretained.
Then
It's not interacting with the stationary phase.
other organic compounds which are maybe polar, less polar, very less polar, whatever may be, they will interact with the stationary phase. So when we talk about organic compounds, mostly we say whenever we are separating compounds, these compounds tend to have certain functional group. And if the compound has certain functional group, then it is true that they will have some kind of interaction with silica. It may be more or less. More means if compound is polar, then the interaction will be more. If the compound is non-polar, then interaction will be less. Okay.
Okay, sir.
Now, if a
Okay,
compound is like when we are using silica as a stationary phase, which chromatography we are doing? Normal or reverse phase?
So normal phase.
Very good. We are doing normal phase chromatography. So on a normal phase chromatography, the compound which is moving fast is non-polar. And the compound which is moving slow is polar. That means there is different rate of migration of the compounds. Got it? Some compounds are moving fast, some compounds are moving slow. And this concept in chromatography is known as differential migration. Known as differential migration. And it is the basic concept of separation. If there is no differential migration, there will not be separation.
Sir. Hello, sir.
Yes, I can hear you.
Sir. Okay, sir. Sir, you were saying the compound that moves fast is polar.
No. It is non-polar. Non-polar.
It's non-polar. Okay, sir.
Yes.
Okay, sir. Now I get it.
You got it. Okay. Now, how to represent it theoretically, right? Because every time I may not be able to give this picture in anywhere, but I can write these results. So retention factor is a way of writing a result, that how the compounds are retained strongly or weakly. Okay.
Okay, sir.
And
Okay,
to
sir.
calculate that factor, we have developed one formula that is called RF. That is equal
Retention
to
factor.
distance traveled by the solute divided by distance traveled by the solvent. Now solvent will always travel fastest. So
Yes, sir.
solvent always will have the maximum distance traveled. Like here you see in this case, the solvent has traveled around 6.2 centimeter. Okay.
Okay, sir.
But the compound you
Okay, sir.
see, they are not traveling as fast as the solvent is. Right? You got it?
Okay, sir. Yes,
So
sir.
we
Yes, sir.
can calculate RF value based on based on the distance traveled by the solute divided by distance traveled by the solvent. So the RF
Okay, sir. Okay,
value
sir.
should always be greater than 0 and should be less than 1.
Okay,
RF
sir.
value will be 0 when the solute remain on the stationary phase at the
Yes, sir.
loading point and immobile.
Fine.
Means suppose if again I go to this slide, if I have loaded something here, if it stays here, so it will be
Yes, sir.
0 by something, 0 by 6.8, 6.2,
Yes, yes, sir.
and it will be 0. That means RF value is 0.
0.
And when RF
Okay,
value
sir.
is 0, that means the compound is not moving. That
Not
means
moving. Okay, sir.
that means the retention is weak or strong.
So the retention is strong.
Retention is very strong. So we have to
Yes, sir. Yes,
weaken
sir.
the retention in this case. So how we can weaken the retention? To weaken the retention, we have to enhance the strength of the mobile phase.
Yes, sir.
Okay. Now suppose if I am running this plate in petroleum ether, then most of the compound may stay here because they are not moving because they are strongly absorbed in that case. But if I run the
Yes,
same
sir.
plate in chloroform, then most of the compound they will go on here.
Move. Okay.
And if
Yes,
I
sir.
run the same plate in methanol, then most of the compound will come here. Right?
Yes,
So we
sir.
have to standardize the
Right,
mobile
sir.
phase according to the polarity of the solutes. Okay.
Okay, sir. Okay, sir.
So RF value will be 0 when the compounds are strongly retained. And RF value will be 1 when it will be 1. Say 6.2 divided
Yes,
by
sir.
6.2, it will be 1. Means all compounds are going on the top. So RF value should always be greater than 0 and should always be less than 1 for
Then
an
1.
ideal
Okay, sir.
separation. Okay. You
Okay,
got
sir.
it?
Okay, sir.
Okay.
Yes,
Now
sir.
tell
Yes, sir.
me something more. You tell me what else you want to ask. I hope you understood HPLC.
Yes, sir.
So HPLC is a closed column chromatography or open column chromatography?
Sir, close column.
Closed column. So it is a atmospheric pressure chromatography or a pressure chromatography?
Sir, it's a pressure chromatography.
Pressure. Very good. Now, what is the full form of HPLC?
Sir, high performance liquid chromatography.
Okay. So why not high pressure? You know, initially when HPLC was discovered, it was called high pressure.
Yes, sir. Yes, sir.
Right. Like you can see this slide, I think. It is called open column or atmospheric pressure
Yes, sir.
chromatography. So
Yes,
where
sir.
we have like the solvent or the mobile phase is moving under the influence of gravity.
Yes, sir.
Right. So when like
Right, sir.
we developed, but this kind of chromatography are very poor with very poor resolution. Why very poor resolution? Because what happens actually, you might have seen this, you can do this exercise in your lab. You take a column and then load the column with say sand, take sand from outside, outside your classroom,
Okay, sir.
and then percolate it with water. You will find that water is
Okay, sir.
not moving.
Yes,
You
sir.
take another column and now you put bigger gravels, bigger like
Yes, sir.
particle size
Yes,
is
sir.
more, and you'll find that water is moving. Now, what
Yes, sir.
happened actually that initially when we started separation, the separation was not good. So we tried that how separation can be improved. And one idea for improvement of separation was that if we enhance the number of retention sites in the column, the separation may enhance. Now, suppose you have a column which is of 2 feet length, and you are putting say 20 gram of stationary phase in that. Okay. Now, if you want to increase the
Okay, sir.
retention site, what will come to your idea? How to enhance the retention sites? Where are the retention sites? The retention sites are on
Sir,
the stationary phase.
stationary phase.
And
Yes, sir.
higher the number of retention site, better is the separation.
Yes, sir.
So how to enhance the number of retention sites?
So we can use the smaller molecules of the stationary phase.
Hmm?
The smaller the molecules, the more they will be packed in a column.
Then what will happen? If the molecule are smaller, then what will happen? Retention site will increase or decrease?
So retention site will increase.
How?
Because there will be larger number of molecules, and each molecule have their own retention sites.
No, you explain it. Your
And so
answer is correct, but your explanation is incorrect. So if I have a particle size of say 1 centimeter diameter, and suppose that particle is having 10 retention sites.
Okay,
now,
sir.
And now suppose in the column there are 100 particles. So that means total retention sites are how much? How many? 1000.
1,000. Yes, sir.
1000. Now I want to increase the number of retention site to 10,000. Means 10 times I want to
Yes, sir.
increase. So what I have to do?
Yes, sir.
What I have to do?
So we can add more molecules.
More molecules. Very good. It's a very reasonable answer. But then what will happen? Means 10 times you want to add the molecules. So suppose
Yes, sir.
in this 20 feet column, you were using 20 gram silica, 20 gram stationary phase.
Yes,
Now
sir.
what will happen? If you add 10 times more means how much?
20.
How much? It's very simple. 200 grams.
Yes, sir.
So if you want to put 200 grams stationary phase, then what problem you will face here? What you have to do?
So we have to increase the column length.
Very good. You have to increase the column length. So initially the column length was 2 feet.
Yes, sir.
Now if you keep the same diameter, then the column length will become 20 feet.
20 feet, yes, sir.
Now can you handle a 20 feet column?
No, sir.
Because what is the ideal or normal roof length in a laboratory? Roof height?
7 to 8 feet.
No. It is around 10 to 12 feet. Minimum around 12
Hello?
feet, 13. It is around 12 feet.
Okay. Okay,
7
sir.
to 8 feet तो कुछ भी नहीं हुआ ना.
Okay, sir.
7 to 8 feet तो अगर आप भी
Yes, sir.
हाथ उठाओगे तो आप भी 7 फुट पे पहुंच जाओगे.
Okay.
ठीक है.
Yes, sir. Yes,
तो
sir.
आप अगर 20 फीट का
10 to
कॉलम
12
लोगे
feet.
तो 20 फीट का कॉलम कहाँ रखोगे आप? It is
Yes, sir.
practically
It will
impossible
not
to
be
do.
possible.
It is not possible.
Impossible. Yes, sir.
So what will be the other solution? What will be the other solution? Think of, and you will understand HPLC. The other solution was, what was the size of the particle I told you? It is 1 centimeter. So 1 centimeter is the
1
diameter of the
centimeter.
particle.
Yes, sir.
And you have put 100 particles, and the total volume is 20 grams, and the length of the column is 2 feet, right? And
yes, sir.
And it is having 1000 retention sites.
Yes, sir.
Now I want to increase it to 10,000 retention sites. So what I can do?
Yes, sir.
Either I can increase the volume by 10 times, or I can reduce the size 10 times. If instead of having a 1000 micrometer, let me convert it 1000 millimeter. Let me convert the 1 centimeter particle size to 1000 millimeter, right? Hello. Hello. Can you hear me?
Hello.
Can you hear me? Yes, Samra.
Yes, sir.
So how many millimeters we have in 1 centimeter? Yes, Samra.
Hello. Yes, sir. 10 millimeters.
You are incorrect. How many millimeters you have in 101 centimeter? Okay, you said 10 millimeter. No, it's correct. Huh?
Yes, sir.
10 millimeters.
Yes, sir.
Okay. So you have 10 mm. Now, if I
Okay, sir.
can reduce the particle size from 1 centimeter to 1 centimeter means it is 10 mm.
Yes, sir.
Okay. Now, if I reduce it to say 1 mm, it is reduced by 10 times.
Yes, sir.
Now, number
Yes,
of
sir.
particles can be increased to 10 times.
Yes, sir.
So there were 100
Yes, sir.
particles. Now it can accommodate 1000 particles.
Okay,
Now
sir.
what has changed?
And they have the same retention sites.
They have the same retention site.
Okay, sir.
No. But
Okay,
now
sir.
retention site has increased 10 times, 10,000.
Yes, sir.
But what is
No,
same?
no, sir. On the one molecule, there will be
On
10
one more,
retention
retention
sites.
site will be same on
Okay,
one
sir.
molecule.
Okay,
Now
sir.
what has changed? Number of retention size has increased to 10,000. But
Yes,
what
sir.
hasn't changed?
So the length of the column.
Length is also not changed and volume is also not changed.
Yes, sir.
Means
Yes,
in the
sir.
same 10 feet, 2 feet column, you have
Yes,
increased the
sir.
number of retention site to 10,000.
Okay, sir.
Right?
Okay,
But
sir.
what problem you will
Right, sir.
face here? What problem you will face here? When the particle size has reduced, there is a flow resistance. The gravitational force is
Yes,
not
sir.
able to move the liquid now.
Low. Okay, sir.
That
Yes,
means
sir.
you are not able to do atmospheric pressure or open column chromatography. So what you have
Okay,
to
sir.
do? Because if the mobile phase stops moving, do you believe that
Yes, sir.
chromatography is possible?
No, sir.
No, because for a chromatography to happen, we need to have a biphasic system. One is stagnant,
Yes,
another
sir.
is mobile.
Moving.
Now here
Yes, sir.
your mobile phase has become stagnant, so you have both stagnant phase and you cannot do chromatography.
Yes, sir.
So how to make your mobile phase moving? How to make it mobile?
So we have to apply the external pressure.
Yes, very good. So we have to apply external pressure. And in doing so, initially we developed one technique called MPLC, medium pressure liquid chromatography. And then finally we developed a technique called high pressure liquid chromatography. Okay. And you'll be surprised to know that in a HPLC column, the size of the particle is 5 micrometer, around 5 micrometer. So you can imagine that how much size we have reduced.
Yes, sir.
And how much, like,
yes, sir.
10, maybe around 1 lakh or 10 lakh retention sites, or
yes, sir.
1 crore retention sites we have generated. So that
Yes,
means
sir.
we have, if you see the same calculation, that 1 centimeter particle, 10 retention site, 100 particle in a column. We have 1 millimeter
Yes, sir.
particle, again we got 10,000 retention site. So 1 millimeter, if I make it 1 micrometer, so it is 10,000 into 1,000, right? So it
Yes,
is
sir.
around
Yes,
1
sir.
crore retention site, but length of the column is same. So you see how much
Yes, sir.
efficiency I have increased the column.
Yes, sir.
And initially the word came to remove mobile phase in this kind of column, I need to apply very high pressure. So if I'm having 1 millimeter particle, maybe I'm applying a pressure of around 50 bars. So this technique was called it was called medium pressure liquid chromatography. And to in a particle of around 5 micrometer, you know what is the maximum pressure HPLC can bear? You have worked on HPLC. You might have observed when you were doing some experiment. What is the normal pressure you can see? The pressure you can see in the column. Do you remember?
No, sir.
So generally, like the HPLC, routine of HPLC in the laboratory to operate from a pressure of around 60 to 100 bars, right? 60 to 100 bars. Okay.
100 bars.
It happens. But yes, depending on the mobile phase, it may increase, right?
Okay, sir.
Like if you are using 100% water, it will be more. If we are putting, say, because water is a viscous solvent. If we are putting 50% methanol, then it will reduce, because methanol is not as viscous as that of water. Okay.
Water. Yes, sir. Yes, sir.
Got it?
Yes, sir.
Okay.
Yes, sir.
Okay then. So So in an HPLC column, we have increased the retention side to around 1 crore and we have enhanced the performance. But pressure word was misleading actually. Initially, two words came. One is MPLC, you can write down. That is called medium pressure liquid chromatography, and another is HPLC, high pressure liquid chromatography. So a pressure of up to 50 bars was termed as medium pressure. And from 51 bar to around 300 bars, like the machines we have in the market at present. So that was called high pressure, right?
High pressure.
But it
Yes,
was a
sir.
misleading word. Like we say that 50 is medium and 51 is high. That was misleading. So instead of and the word HPLC became very popular. So the word pressure was replaced by performance.
Performance.
So
Okay, sir.
the abbreviation remains the same. HPLC is the same.
Same.
The word pressure is replaced by performance. Why we replaced it by
Performance. Yes, sir.
performance? Why we replaced by performance?
Still,
Because when the
because it
pressure is
generates more
increasing.
output.
Yes, no.
Yes, sir.
You understand why the pressure is increasing? Because we are reducing the particle size. And when we
Yes,
are
sir.
reducing the particle size, the separation is improving.
Proved.
That
Yes,
means
sir.
the performance of the system is improving. So based on
Improved.
that,
Yes,
we
sir.
said that the performance is enhanced. So that's why we call it that we have a high
High
performance
performance.
liquid
High
chromatography.
performance. Okay. Yes, sir.
Okay, very good. So you got it, I think.
Yes,
So
sir.
I think this can you see this slide? I
Yes,
just
sir.
want to show you this graphic, and you see this is a modern HPLC machine where we have a
Yes, sir.
pumping system, because this is the column, and the column in an HPLC is made up of, you might have seen it.
Yes, sir.
It is made up of what material? It is made up of stainless steel.
Stainless steel. Yes, sir.
So that it can bear the high pressure. If it is a glass
High
column, it may
pressure.
break under pressure.
Break.
Right?
Yes, sir.
Right. And so
Yes, sir.
to pressurize solvent in this system, we need to have a mechanical pumping system. So we have pumping system. So these pumps, like there are based on the number of pumps and number of solvents you are using. We have isocratic system, we have gradient system, we have in gradient, we have binary gradient, ternary gradient, and quaternary gradient. So this is a binary gradient system where we are
Okay, sir.
using two solvents. Okay. So these two solvents,
Okay,
these
sir.
two pumps, they are mixing the solvent here and they are pumping the solvent to the column. Because since from pump to column, pump to detector, it is a pressure system, under pressure system, right? So how to load the sample?
Yes,
So
sir.
we have a pressure injector here, inline injector, where we can inject the sample inline. The sample will go to the column. Whatever chemistry we have in the column, whether normal phase, reverse phase, or any other chemistry, the sample will be
Okay, sir.
separated. Now, how to detect the sample? So for that, in HPLC, we have online detectors. So different
Yes,
detectors
sir.
we have. The most common detector is a UV visible detector, which detect the
Okay, sir.
compound based on its lambda max, right? The other detectors are like IR detector, NMR detector, mass detectors, everything I have given. So you now just see a graphic, how it work actually. And you see it very carefully. See, we have loaded a sample. Can you see it here? Can you see it here?
No, sir, it's not moving.
Oh, it's not moving. Wait. I think there is a huge trouble at your end. Now, can you see it?
Yes, sir.
Can you see it now? Can you see the sample which
Yes,
is
sir.
near my cursor?
Yes, sir.
Now
Yes, sir. Yes, sir. The pink color one.
the sample has been loaded here. Now it
Yes,
is
sir.
a graphical representation because everything is made up of steel. You cannot see anything. Only thing
Yes, sir.
you can see the
Is the
output
chromatogram.
of
Yes,
a
sir.
detector. So this is a detector.
Yes, sir.
So when there is no sample in the detector, you see there is a baseline which is at 0. Can you see this baseline?
Yes, sir.
Can you see it? So this
Yes, sir.
baseline is at 0. That means nothing is coming to the sample to the detector because no sample is like coming out of the column. The sample is here. Now you see the sample is going.
Okay, sir.
It is loaded in the column, and you see when it is loaded in the column, depending on the chemistry of the column, the sample is separated. Can you see that? You can see different bands in the
Yes,
column.
sir.
Okay, this is a
Yes, sir.
graphical representation, mind it.
Yes, sir. Yes, sir.
But still you do
Yes,
not
sir.
see anything. The baseline is at 0. A small notch
Yes, sir.
we generally see,
Hmm.
because when we
Yes, sir.
rotate this injector port, then there is a disturbance in the flow of mobile phase. So this notch is generally by the flow of mobile phase, right?
Okay, sir.
Now you see here. Now you can see that there are 1, 2, 3, 4, 5, 6 compounds are there, which are being separated in
Yes,
the
sir.
column, but still you see the baseline is 0. Why the
Yes,
baseline
sir.
is 0? Because
Sir,
nothing
it's
has reached the
yes, sir.
detector. So
The
there is
detector.
nothing to
Yes, sir.
detect. Now you see very carefully, the first compound is reached the detector,
Yes,
right?
sir.
You see the first
Yes,
compound
sir.
is reached the detector. Now when first compound has reached the detector, you can see a peak.
Yes, sir.
Okay. Now you see this compound is in the detector, you can see a peak. And the second compound is going to the detector. Now
Yes,
you
sir.
see the first
The second
compound
peak.
is
Yes, sir.
detected, and the second compound in the detector, you can see the second peak. Now
Oh,
you
yes,
see
sir.
the third compound is coming, but now you see the distance. Now you see the distance between first and second compound is very less, right?
Yes,
So
sir.
you see
Yes, sir.
that the two peaks, they are separated, but the separation is it is okay separation. It's not bad, but it is okay. Now you see the third compound is now going to the detector, and you see
Yes,
the
sir.
third peak is coming after a huge difference. Like you see
Huge
the
difference. Yes,
difference
sir.
between RT difference between this and this is very high, and RT difference
Yes, sir.
between this and this is very low. That means
Very
their
less. Yes,
compounds
sir.
are having structure which is matching with
Similar.
each other, similar structures, or similar polarity, but
Yes,
this
sir.
compound is having different polarity, right?
Yes, sir.
Now the
Yes,
fourth
sir.
compound is going to the column. So this is fourth, fifth, and sixth. Now you can see
Sixth. Yes,
fourth,
sir.
fifth, and
Fifth and
sixth, and
sixth
you can
and
see all
sixth.
compound.
Yes,
Now
sir.
you can see 1, 2, 3, 4, 5, 6
4,
compounds are
5,
detected.
6.
Okay.
Yes, sir.
So this is a
Yes,
chromatogram.
sir.
On
Yes, sir.
x-axis you have time. This is called retention time. It tell you that when which compound is coming out of the column.
Coming out.
So you can say
Okay, sir.
this is the compound which is having highest or lowest retention.
Sir, it has the lowest retention.
Very good. This has the lowest retention, and it will have the lowest RT, maybe say around 3 minutes.
Yes, sir.
That is 4 minutes.
Yes, sir.
That is 6 minutes. It is 8 minutes, and say it is 15 minutes. So this compound,
Yes,
or
sir.
say it is 20 minutes. So this compound is having the highest retention.
Highest
That
retention.
means its polarity is matching with the polarity of the column. If the column
Yes,
is
sir.
C18, this is a non-polar compound. If the column is silica, this is a polar compound.
It's a polar compound. Okay, sir.
Right?
Right, sir.
So you got two information from here. That one is quantitative and qualitative information that tell you that how many compounds are present. You can say that in this mixture 6 components are present, right?
Yes, sir.
On y-axis you see milli absorbance unit, MAU.
Yes, sir.
MAU stand for milli absorbance
Milli
unit.
absorbance unit.
Just a second. I'm getting a call. I just call you after 2 minutes.
Okay, sir.
Okay. Yes, Samra, can you hear me?
Yes, sir.
So now you see that we got two details. One detail is like our
The
milli
qualitative.
absorbance, matlab milli qualitative means
Yes,
how
sir.
many components are present.
Hmm.
And on the
Yes, sir.
y-axis, you see this is milli absorbance. So
Yes,
how
sir.
much is the absorbance tell you about the quantity of the component.
Yes, sir.
So you get quantitative
Okay, sir.
information. Like you can see that this peak
Yes,
is
sir.
having the highest peak. That means this component is having the
Mm-hmm.
maximum concentration in the sample.
Yes,
And this
sir. Yes,
component
sir.
is having the
Least
lowest
concentration.
concentration. So these detectors, they are online detectors. They detect the component simultaneously. Okay. Means
Yes, sir.
compound is coming detected, and we get a chromatograph. Okay. So I hope
Okay,
you
sir.
got the idea that how this
Yes,
chromatography is
sir.
performed. Can you see the next slide?
Yes, sir.
Now you can say
Yes, sir.
this is time called, this time is called T0. T0 means when the sample was injected,
Mm.
and then we can calculate the
Yes, sir.
TR, time retention of every compound. And
Every
based on
compound.
that
Okay,
time
sir.
retention, we can find out that whether the sample is separated properly or not. This we discussed, right? And these are the common detector that has been
Yes, sir.
used. UV visible is the most
Mm.
common detector. Can you see the slide? This slide,
Yes, sir.
detector slide.
Yes, sir.
Then UV visible, fluorescence, IR, mass, or NMR. These are the common detector which is used with the HPLC. Okay. Now,
Okay, sir. Okay, sir.
advanced to HPLC, we have another type of chromatography that is called UPLC. That is called UPLC. Can you read it? What it is written? Can you read
It's
what is
ultra
written
performance
here?
liquid
It is ultra
chromatography. Yes, sir.
performance, right?
Yes,
Ultra
sir.
performance.
Ultra performance. Yes, sir.
So I have told you that HPLC routinely they perform up to 3,300 bars. Now UPLC can perform up to 1,000 bars.
Yes, sir.
The particle size in HPLC, what I
1,000
have told
bars.
you.
Okay,
The
sir.
particle size, what I have told you. Particle size in routine HPLC column is 5 micron. How much?
5 micron.
5
Yes, sir.
micron. And
5
here it is
microns.
reduced to 1.7 micron.
1.7 microns.
Almost it is say around like more than 2 times it is less. So the
Yes, sir.
performance
Yes,
is
sir.
increased. Now you compare like in a routine HPLC program, the runtime was 30 minutes.
Yes,
Okay.
sir.
So the particle
Yes, sir.
size is, you can say it is 5 micrometer. Huh?
Yes, sir.
Now here you see the runtime is how much? 5 minutes. Can you see it? 5 minutes.
Yes, sir.
So
Yes,
the
sir.
work which we were
5 point. Yes, sir.
completing in 30 minutes can be completed in 5
We
minutes.
can do it in 5 minutes.
So it
Yes,
is
sir.
called ultra performance, right?
Ultra performance. Okay.
Okay. So I think these you can read, and in case
Yes,
if you
sir.
have any issue, just let me know. And this is
Okay, sir.
because you have the gas chromatography. I just want to discuss for a while with you about the gas chromatography. So in
Okay, sir.
gas chromatography, the mobile phase is gas, and there is a difference in
Yes. Yes, sir.
this is the like the what do you call it, the instrumental design, where you have a gas at high pressure, then it goes to a column, and the column here you see it is a coiled column, and the
Coiled
length
column.
of the
Okay, sir.
column in GC is around 100 meter. So imagine a 100-meter column. So it is packed inside
Okay,
a
sir.
oven. Why it is packed inside a
Okay,
oven?
sir.
Why it is packed inside a oven?
So because we have to provide it the optimum temperature to keep it in a liquefied state.
So it is a yes, we have to keep it always in the keep it
Liquefied
always in
state.
the liquefied, maybe vapor state, not liquefied.
Yes, sir.
You have to keep it
Yes,
in
sir.
vapor state. So if we have to keep it in vapor state,
Vapor
then
state.
we have to keep it always heated, right?
Heated. Yes, sir.
Right. Huh? Okay. So this is one difference between this chromatography and HPLC, right? Okay, so and separation takes place, and the detectors used in this one are, one is flame ionization detector, because these like the compounds are in vapor state, so the compounds are detected in vapor state only. Okay,
Hmm.
so in one detector,
Vapor
what
state.
happens, like the
Okay, sir.
compound which is coming from the column in vapor state, it is burnt in flame. So when it is burnt in flame, the compound
Okay.
disintegrate, and when the compound disintegrates, the electrons are liberated. Now these liberated electrons are
Okay,
collected
sir.
and given to a circuit which generate a current. So you have a chromatogram
Okay, sir.
of time versus, you can see it is written volt, time versus current.
Volt. Yes, sir.
So in HPLC,
Okay, sir.
the chromatogram was time versus
So the absorbance may
absorbance,
absorbance unit.
because it was a UV visible
Yes, sir.
detector. Now here
Yes, sir.
it is time versus voltage, time versus current.
Voltage. Voltage.
Okay.
Yes, sir.
Okay, so we get
Okay,
this
sir.
information, and then we get the information about the peak number, retention time, area, and then percentage area, like this. Okay, so we have three
Uh-huh.
kind of detectors. You just read those detectors in detail, and in case if you find any difficulty in understanding anything from this topic, then you just let me know. Okay,
Okay, sir.
now do
Okay,
you
sir.
have some questions? We have around 4 or 5 minutes, we can discuss it.
Sir, I was going through it and I found one thing, that was gel filtration and gel permeation chromatography.
Yes, I think that is not there in your syllabus, but I have shared the complete chromatography presentation with you.
Okay, sir.
So
Okay,
gel
sir.
permeation or gel
I don't
filtration
know actually what's in my syllabus.
is a size
I was
exclusion
going through the
chromatography.
slides.
Yes, yes, you can read it because there is no limitation to reading, na?
Yes,
And
sir.
it is a
Yes,
very
sir.
important technique actually,
Yes, sir.
because for in biology we do not
Okay,
use
sir.
much of GC, but we use lot
Okay,
of
sir.
gel permeation or gel filtration. The total like
Yes,
protein
sir.
or DNA work for separation of protein, we always use gel permeation chromatography. That is where
Yes, sir.
there the separation is based on size. You know what
Hmm.
happens
Yes, sir.
actually? I think I have told you that protein is such a complex molecule that it is very
Yes,
difficult to
sir.
separate protein molecule based on polarity,
Hmm.
because there will
Yes,
be
sir.
huge, there will be very minute difference in polarity of two molecules. So it will be very
Yes,
difficult
sir.
for separation. But yes, there may be huge difference in their size. So it is very easy to
Okay,
separate
sir.
protein based on size. So we have this chromatography, size exclusion chromatography, or we call it
Yes, sir.
gel permeation chromatography, or we call gel exclusion chromatography, in which the protein is separated based on the size.
Size.
Okay. So you read
Okay,
all
sir.
the total this one, and you just send me one WhatsApp message, hello, on WhatsApp, my mobile number, and I will
Okay.
share
Okay, sir.
you with the electrophoresis presentation today. And in our next
Okay,
class,
sir.
we will have, tomorrow we have class.
Yes, sir.
So tomorrow I will start discussing with you about the electrophoresis. And I wish that in
Okay, sir.
Saturday, Sunday, you read about electrophoresis completely, so that in the next
Okay,
class we
sir.
will discuss it. Huh? And I will
Okay, sir. Okay,
share
sir.
it on your mobile number, because I think nobody is sharing it on the group. So I have shared this one on the group. So I will share
Yes,
it
sir.
within your mobile number. You read it, because I have prepared this literature only for the
Okay, sir.
students to read, and I share it very freely
Okay,
with
sir.
my
Okay,
students,
sir.
so that they can read it.
Okay, sir.
But my purpose is that you should read,
Okay, sir.
because unless and until you read, you
Yes,
will
sir.
not have any questions. And if you do not
Yes,
have
sir.
any questions, you will not be able to understand the concept, right?
Yes, sir.
So to understand the
Yes,
concept,
sir.
you must read it properly.
Yes, sir.
And you should come up with a lot of questions.
Okay, sir.
I find you are asking less questions. You should come up with a lot of questions.
Okay, sir.
Okay, and for that you have to
Okay,
read
sir.
my, because I think I have shared you around 80 slides.
Yes, sir. No, no, sir, there are 100 slides.
100 slide, whatever it is.
Yeah.
So you just read it, and then you will come
Okay,
up
sir.
with the queries, and then you can ask me in my next class. Okay.
Okay, sir.
Okay
Okay,
then.
sir.
So
Okay,
I'm
sir.
stopping here.
Okay, sir.
Okay then.